Hello, everyone. A very good morning and good afternoon to all of you joining us live today. And hello to all of you who will listen to us offline mode later on. So this webinar will also be recorded and accessible for offline viewing. So I would like to start with a very warm welcome to all of you on our webinar. My name is Bhumika Singh from Kirkstall Limited in the UK. And I'm very pleased to introduce our special guest today, Dr. Doris Wilfing-Cedar, who's joining us from Austria. Doris is an associate professor at Medical University Innsbruck at Austria. So Doris today will talk to us about the fast-track development of lung and immune cell model. This is an in vitro 3D model that was developed in her lab, utilizing the Kirkstall's Quasi Vivo dynamic flow system. So before Doris takes over, I'm going to give you a little background on the Quasi Vivo concept, the different Quasi Vivo systems, and application examples. And in the end, both Doris and I would love to take any questions that you may have. And also for your information that for people who are joining us live, you can also submit your questions on the control panel that you can see on the screens. And we'll make a note of them and try and answer as many as we can in today's session. So, moving on to the concept of Quasi Vivo. So we know that Quasi Vivo systems are actually enable milli-fluidic cell culture with controllable flow rate. It is known that most cells in the body are exposed to fluid flow. The schematic on the screen shows the interstitial flow crossing an artery wall, exposing smooth muscle cells and fibroblasts to fluid shear stress. It is known that interstitial flow regulates the microenvironment of the tissues and many physiological responses. However, most in vitro modeling studies do not include these important physiological elements in their models. The gap between the in vitro and the in vivo models remains unaddressed. The predictability of current preclinical studies is low. These lead to high failure rates, costing the drug discovery industry astronomical sums at late stage of clinical trials. The predictability of clinical outcome could be increased if in vitro models include some of the key physiologically relevant aspects such as flow and dynamic interactions between different cell types of the body as well as the microbiome. Quasi Vivo systems are designed to address these two main issues. So on the slide here, you can see all of the three Quasi Vivo systems, QV500, 600, and QV900. All of the three systems allow precisely controllable media flow through a peristaltic pump. All the three systems enable modular cell culture. This means different tissue culture units could be interconnected, creating multi-organ physiological systems. These are increasingly becoming important in pharmacodynamic, pharmacokinetic, and disease modeling studies. As I said earlier, that there are three Quasi Vivo systems available, and depending on the application, the flow rate could be set at a required setting. Within a system, one or more flow circuits could be easily generated. Each flow circuit could be set up with different flow rates. For example, a model of liver with relatively lower flow rate connected to a vascular model with a higher flow rate. So the Quasi Vivo 500, 600, and the 900, they all support fluid flow, submerged cultures which are modular and could be easily connected. All the systems are designed to eliminate air bubbles. QV600, which we are going to talk about today in much more detail, and Doris is going to explain to us how she utilized QV600 in her lung immune cell model. I'm going to give a little bit more information as to what additional features this model has. So QV600 has some additional features that it can support 2 chambers and can also support air-liquid interface. The QV900, on the other hand, has 6 disposable units on a standard well plate format, which you can see on the right side of the screen. And this is a compact solution for multi-organ or organ-on-a-chip approaches. On the next slide, I have listed the benefits of flow. So why do we need flow in the in vitro environment? Definitely it delivers oxygen and nutrients to the cells. It removes waste products, stimulates cells, because cells do require flow or shear stress. It provides relevant mechanism for cell-to-cell signaling and enables systemic multi-organ models approaching. So researchers in the University of Pisa have shown that flow rate in the Quasi Vivo chambers is of laminar nature. The flow rate increases as we go from the bottom of the chamber to the top of the chamber. And Quasi Vivo systems allow adjustable flow rates depending on the in vitro application. In vitro models on Quasi Vivo system are biomimetic and with greater clinical relevance than conventional static models. This makes the QV, the Quasi Vivo, fit in the preclinical stage of the drug discovery where highly predictive efficacy and toxicity evaluation are required. So because of the increased clinical relevance, Quasi Vivo systems are becoming increasingly popular in the laboratories globally, with majority of users in the USA and Europe. So there are peer-reviewed publications from Quasi Vivo users in diverse application areas. Today, Doris will present the respiratory model that utilizes the Quasi Vivo 600. So a little bit of more detail as to the technical aspect of the Quasi Vivo 600, which you can see on the middle of the screen. So this unit fits any standard transwell insert. It has tubings for flow to the top and the bottom of the compartments. And there are two compartments, so the tubings connect to both the top and the bottom compartments, which could be used to create two separate flow circuits. So if you want an air-liquid interface, your setup would look as shown on the left side of the screen. So in the air-liquid interface model, the media is flowing only in the lower compartment, and the tissue model growing in the upper compartment could be treated topically or with airborne contaminants, as could be the case with your models in the application area of lung or respiratory model, or in the case of skin, where topical applications could be delivered. On the right-hand side is the liquid-liquid interface model, where media is flowing through both upper and lower compartments. For example, cut epithelium model that could be treated with cut microbes. In the lower compartments of both the models, both the air-liquid interface and the liquid-liquid interface, additional cell types could be cultured, either at the bottom of the chamber or on the reverse side of the transwell membrane. Thus, this advances the physiological relevance because we can include more than one cell type and study interactions between the different cell types. So that's about me for the Quasi Vivo 600 setup. I would now hand over to Doris, who will explain in detail the lung immune cell model using the QV600 system. So please welcome Dr. Doris Wilfing-Sila.
Hi, Bhumika, dear users of the Quasi Vivo, dear listeners. It's my pleasure to show you today our data on fast-track development of an in vitro lung immune cell model to study Aspergillus infections, which was published last year in Scientific Reports. So I also want to thank you for the nice introduction and the nice overview on Quasi Vivo systems, because this makes my life much easier. Today I want to show you what we are working on. So we are interested in host pathogen interactions and especially also in pathogenic fungi. And you have to imagine that in healthy, normal individuals, fungi don't do much, because otherwise we would be exposed to pro-inflammatory reactions all the time, because we are inhaling thousands of conidia, fungal conidia per day. And what happens when we inhale them, and when we are healthy, then the conidia are first trapped within the mucous layer of the lung or respiratory epithelia. But whenever the conidia are able to break through the borders of those epithelia, the immune cells are immediately activated and reacting on incoming pathogens. And in this case, the most important immune cells are alveolar macrophages, dendritic cells, or also neutrophils. But what's the situation in immune-compromised individuals? In immune-compromised individuals, the immune component is missing or very weakened, and therefore the fungal pathogens are able to enter the epithelia and are able to form hyphae within the lungs. But this, as said, is only happening whenever we are immune-compromised. And that's why we are also interested in pathogenic fungal infections, because there is a massive increase in the number of fungal infections, especially for Aspergillus fumigatus, due to the rise in the number of immune-compromised individuals, which, for example, undergo hematopoietic stem cell transplantation or who are also immunodeficient, for example. And besides fungal virulence factors, there are also multiple predisposing factors to be studied, such as the role of immune cell, antifungal resistance, or also within mixed population. And there's really a need for a potential true-to-life 3D respiratory model using primary cells. And this is what we set up in our lab. So we set up a perfused 3-dimensional in vitro cell culture model consisting of either normal bronchial humoral epithelial cells or small airway epithelial cells. And then we added immune components such as macrophages or dendritic cells. And now I want to show you why we decided to take macrophages or dendritic cells. The dendritic cells, you have to imagine, those are really the sentinels of our immune system. So these are really the one and only cells of our immune system which are able to activate naive T cells to become either Th1, Th2, Th17, or T regulatory cells. And these are the only antigen-presenting cells able to do so. Not macrophages or B cells are able to activate naive T cells. And since we are also interested in the T cell response, we decided to work on the one hand with dendritic cells, but on the other also with alveolar macrophages, because these are the first immune cells which are confronted with fungal pathogens, since they are sitting on the apical side of the alveoli and the small airway epithelial tracts. And that's why we decided to start with these cells first. And now I give you just a short overview. What are the current approaches in 3-dimensional cultures? So on the one hand, you can do organotypic explant cultures, you can do cellular spheroids, you can do hanging drops, you can do polarized epithelial cell culture, which we did, or you can also take artificial skin or microcarrier cultures. And we did take the Transwell cultures, and for this we used primary cells. On the one hand, normal human bronchial epithelial cells, which are coming from the upper respiratory tract, or small airway epithelial cells coming from the lower respiratory tract. And these cell types differentiate in the fact that NHBE cells do have goblet cells, which produce the mucus, while small airway epithelial cells, they do also have goblet cells, but in addition, they express the Clara cells, or nowadays called club cells, and therefore we use those two models to add our immune cells there. And for this, we also used the Quasi Vivo 600 system to culture the cells under perfused conditions and to have an interconnected cell culture system. And here you can already see how this looks like. So you have the silicone chambers where you put in your transwell, as Bhumika already told, and you connect this and the reservoir bottle to a peristaltic pump, and then you are able to pump the medium through the system. And the advantages of such a system is that it looks like the in vivo conditions where you also always have a flow. You can also study multiple parameters. You can also by this method reduce animal experimentation, and you also have a batch-to-batch reproducibility. Here you can see how we build up our system. So as you can see, this is the pump. Here we have the pump tubings, which differentiate from those which go into the first chamber. There are the transwells in there, and the flow is just below the transwell, the bottom of the transwells, while in the upper well you have an air phase. And then you can also connect the second chamber, and then the medium is pumped back into the reservoir bottle via a second tubing of the pump. So you can see this is a closed system, and what might also be an advantage here is that media from the chambers are also enriched within the reservoir bottle. And to see whether the system is really of advantage for our cells, we cultured NHBE and small airway epithelial cells, either under static or perfused conditions. And now I want to show you some results from this study, because what we found was that under static conditions, the cells showed a slower differentiation compared to cells which were at the same age in air-liquid interface under perfused conditions. And this we could measure by production of mucus, as shown by MUC5B marking, by cilia differentiation, as shown by beta-tubulin, and by tight junctions, as shown by occludin. And here you can see a merge of all those markers, and you can easily see that under perfused conditions, the cells simply look more differentiated. In the next step, we also looked at the differentiation by scanning electron microscopy. And also here we could see those differences, because under static conditions, you might see that, of course, there are cilia, but it's in a much less differentiation state compared to the cilia under perfused conditions. And this is also shown here. I mean, here's a bundle of cilia, while here you can see really a nice overview on the cilia. Of course, we next looked at the trans and epithelial electrical resistance to see at the intactness of the epithelial membranes, and we measured the tears under perfusion, which is shown here in green, and under static conditions. And on the first days of differentiation, you can easily see that under perfused conditions, the tear is significantly higher under perfused conditions in both NHBE as well as SIE cells compared to static conditions. The tear then gets more similar under static conditions whenever they get older. So whenever it's around day 21 in air-liquid interface, you see about the same tear values under static and under perfused conditions. So it simply takes longer under static conditions, and therefore this is really an advance of the perfused system. Here we did live cell analysis of the cilia under static and perfused conditions, and you can see that, of course, there are cilia here around day 14, but they are very small, and not too many cilia are there. But under perfused conditions, you can see that much more cilia were developed, and they are much longer than those compared under the static conditions. So next, since we are interested in the uptake of Aspergillus fumigatus conidia by respiratory cells, we next did short-term scanning electron microscopic analysis of fungal conidia with differentiating epithelial cells. And what we found that was indeed that the conidia were taken up by the epithelia after 3 to 6 hours. But of course, the epithelia are not really the cells which are combating fungal infections. but these are either dendritic cells or macrophages. This you can nicely see here in this overview, which was done by Ido, Goyer, and Steinman, where you can see that the dendritic cells here sit on the basolateral side of the respiratory epithelia, stretching out their dendrites to catch possible danger signals within the respiratory tract. Or on the other hand, you can also see macrophages on the apical side, so on the air side. So we first added dendritic cells to the basolateral side, and these dendritic cells were stained using CFSE to appear green. And then we added a red label Aspergillus canidia to the system and wanted to see whether the dendritic cells interact with those canidia in the system. And this is shown here. This was shown by confocal microscopic imaging. And here you can see that the dendritic cells already after 1 hour co-localize with the fungal conidia, so therefore they will stretch out their dendrites and catch the conidia. And then with time, since the conidia within this closed system grow out to hyphae, which then also produce toxins, they destroy the epithelium. And by this time, so by 24 hours after exposure to the fungi are grown out to hyphae, they destroy the epithelia, and the dendritic cells come up to the apical side and co-localize with these hyphae. This was not the case in an uninfected control, where we did not see any migration of dendritic cells to the apical side at all. So therefore, in this primary system, you also do not see any unspecific activation of the immune cells, which were added to the basolateral side. So in the next step, we added macrophages to the apical side, so to the air side. And then we again infected our tissue with red labeled conidia, which are shown here. And after 6 to 24 hours, we again see this aggregation of macrophages on the apical side around the conidia and co-localization of the conidia with the macrophages. And after 24 hours, since again the conidia were grown out to fungal hyphae, the conidia, they are really aggregating around the hyphae. But of course, as said, in such a closed system, it's not possible to do some escape from the fungal damage. So therefore, after 24 hours, we had to give up the experiment in principle. So in yellow, you can see here also internalized macrophages, since here you can see the co-localization, for example, of fungal either hyphae or conidia with the macrophages. What we are going on right now is to go towards animal-free in vitro 3D cultures, because all those experiments before were done with coating of the transwells with red tail collagen. And now in the last experiments, which are still unpublished, we are using a birch-based hydrogel from Grodex, which we coat the transwell with, and then we there seed our epithelial cells and then add our immune cell components. And also from these parameters, we have several readout parameters such as TEER, then high content, high throughput screening, live cell imaging, confocal imaging, or measuring cytokines. And as you can see here, in this system, the differentiation of the cells is very far and also within a shorter time point compared to red tail collagen. The cells are growing within snail-like features, and this is also really looking nice. You can also see here a very nice uptake of or a very nice live cell imaging of those highly differentiated tissues. And in future, of course, we also want to apply these tissues into the Quasi Vivo system to see whether this further improves the development of the cells. And here I want to shortly summarize the data we found within our studies. With our systems, we are able to either look at epithelial interactions with pathogens alone. We can look at immune cell interactions with pathogen alone. We can, of course, combine those various cell types and take a look at interactions with those. And we can also add T cells in the bottom of the transwells, and therefore look at also interactions with T cells, which is also a further point within our studies. So to summarize Bhumika's and my talk, we have to say that shear stress is physiologically relevant because in our tissues we also have the shear stress, and we have the ability to closely control this stress with the Quasi Vivo systems, and we can also co-culture multiple cell types and organs. It's an easy-to-use system and also compatible with the standard workflow, and we can also scale it up or down because we have enough cell material for tissue-based and also endpoint analysis. What we found within our system was an accelerated development and differentiation of the airway epithelial cells, as well as an increased efficiency of in vitro models with the Quasi Vivo system. And with this, I want to thank especially Parul Chandorkar, who does her PhD thesis in my lab, and she set up, in principle, the whole system with the help of our senior scientist, Wilfried Bosch. And in my opinion, they did a really good job, and Parul is also now with me to also answer the following questions. And I also want to thank you for your attention. Thanks a lot.
Hey, thank you, Doris. Very impressive presentation. Really, really nice work. So, so here we've started the question and answer session. So, we've got a few questions coming in. So please keep your questions coming in while we answer some of the questions we've already got. So the first one is from Lena Rushi. So his question is the flow rate of Quasi Vivo 900 if we use hepatocytes. Is it 130 microliter per minute? So thank you, Lena, for your question. So yes, so we can, the range of the flow rate can be between 120 microliter to 300 microliter. So we can share more details with you offline. So please connect with us so we can discuss it further with you. Next question I would like to take is about I think it covers the efficiency of the Quasi Vivo system. So the question is, how long does it take between the seeding of the cells, switching on the flow? And the second part of the question is, did any cells detach? So I think, Doris, this question can be for you. Do you want me to repeat, Doris?
Yeah, what?
The first part.
No, I think I got it.
Okay.
I think I got it. Do you hear me?
Yeah. Yes, Doris.
Okay. I think what we did is to keep the cells first for 3 days under submerged conditions, and then we transferred these into air liquid interface into the perfused system, and thereafter about 12 to 14 days we were able to see the cilia under perfused conditions, while under static conditions it took 3 weeks to see the cilia.
Yeah, okay, thank you, Doris. So the second question we have is what medium did you use for your air liquid interface culture?
The cells were obtained from Lonza, we have used the medium that is provided, which is commercially available with the cells from Lanza.
Right. Okay. Thank you, Parul. So, got another question. So, I think it's related to the technical part of the culture. So, how many cells do you seed per chamber?
30 cells per chamber per transwell insert.
Right. So there's loads of questions coming. So I'm just trying to see not to repeat any. So that's why there's a little bit of delay. The next question is from Kevin Kelly. The question is, how do you measure transepithelial resistance, TEER? Is the system opened or do you sacrifice samples? Is there any integrated TEER electrode? So there's quite a lot in there. So let's answer it. Let's split the question. So how do you measure a transepithelial electrical resistance?
We have always used one well, which we used for measuring the tear. So, but we always used the same well. So we just didn't want to bring some contaminations in other wells. So that's why we used one specific well for measuring the tear, and we measured the tear using the EVOM ohm meter. The EVOM 2 ohm meter, and maybe Parul can say some more details.
Yes. So basically, the EVOM 2, the ohm meter has 2 electrodes, and it has the electrode, one which has a longer length than the other, and this is inserted into the transwell with one electrode dipping into the well that is touching the membrane, which has the epithelial cells, and the other which touches the which is inserted into the well. And this is how we measure it, and then we can it is basically expressed in ohm meter square, and that's how we generally measure it.
Okay, a few more questions, so I'm going to keep them short. So the next question is from Shikhar Saha. The SAE cells show less TER than the NHBE. So is there a specific reason, or is this expected?
Why the SAE cells do have less terror? What was the question? Sorry.
So I'm trying to understand. So Shikhar just wrote a statement here that SAE cells show less TER than the NHBE.
Yeah. Mm-hmm. Yeah. I think it's also due to the more mucus the NHBE are producing. So I mean, those are there you really also see a pretty high mucus production, and this one is much lower in SAE cells, and also I think the cells are maybe a bit. No, I think it's mostly due to the higher resistance by the mucus. Yeah, I would suggest.
Yeah. So another part of our question was how physiologically relevant these the relevant flow you've used in the system. So if you were to do it again, would you change anything to the flow environment?
Yes. So the flow rate that we used was 100 microliter per minute, and we have optimized this with our cells. And this is, if we were to set up the system again, this would be the flow rate that we would use in future, because we optimized it for our system.
Okay, good. Few more questions. I'm really happy that there are so many questions coming. So So this question is about the use of the Quasi Vivo system in the model development. So can you comment on how this speeding up the model development, because it's about fast track development of the lung immune cell model. So how use of Quasi Vivo helped?
I think it helped a lot because, I mean, as you have seen from the pics, the differentiation is much faster, and therefore you can start with your real experiments about a week before, for example, starting under static conditions. So therefore, I would say that that it's it's helping save time and also money, because you can start already after about 2 weeks or up to 17, 18 days, and under static conditions you could start after 21 to 28 days or something like that. So therefore, yeah, I think this helps a lot.
Okay. So some of the viewers are experiencing some problem in the line, so please let us know if you can't hear us properly. So if you just indicate on the control panel if the line right now is all right. So I'm going to take the next question from Juan. How often do you change the medium?
So we basically added, so the reservoir bottle usually has 20 ml of the medium, and we usually always top up every week with 5 to 7 ml medium.
Okay.
But the complete medium is not usually changed because the medium, because the whole purpose of having this flow system is to have the conditioned medium in circulation, which aids in faster differentiation.
Good. Next question is, I know you have used the fungus in your model development. So if we were to use the dynamic flow system with microbes, what approaches do you recommend for people who want to use the microbes in the dynamic flow system, especially with fungus?
Yeah, that's, I mean, we had to put them after culturing in air liquid or differentiating in air liquid interface. We had to put them into static conditions since we are working with pathogenic fungi, which, as I've said, overgrow everything within a very short time period. And since we did not want to contaminate the system and also make the tubes somehow blocked. We put the wells afterwards into the static system. But maybe also, if you just want to look at interactions of pattern recognition receptors with, for example, hyphal pathogen-associated molecular patterns, you could also, for example, let the fungi grow into hyphae and then UV inactivate these and add those fungal those pieces of small pieces of fungal hyphae to your medium. This might work. Or if you do have other respiratory challenges, for example, such as nanoparticles or something like that, or also bacteria, you might either inactivate the bacteria or maybe, yeah, you can, of course, can also afterwards do some sterilization or autoclaving, but that's not possible with the pathogenic fungi. But I would suggest it's also possible to add microbes to the system, but maybe not adding sterile cultures afterwards. So I would order 2 systems, one which I would only use for sterile working, and the other one maybe for the work with microbes.
Good. So next question is interesting one from Beatrice Pecoraro. So your question is, do you consider this system as appropriate to conduct and high throughput screening? So, Doris, would you like to take this, or you want me to answer? Maybe we both can. I'll let you speak, Doris. First.
Yeah, I mean, with the QV600, it's a bit difficult. I think the QV900 is good for high content imaging or screening purposes. For the QV600, I still see the problem that yeah, I mean, we only cultured up to 10 slides per experiment, but maybe you say your impression, Bhumika. I mean, that's just what I'm facing, that with the QV600 right now, it's yeah, I mean, of course you can take several pumps, but I think right now it's maybe a bit difficult.
Okay, yeah, so absolutely. So I would like to add just a couple of points in there. So QV600 is not meant for high throughput screening in a nutshell. However, we've got QV900 that also delivers a dynamic flow at a controlled rate. And it's got 6 chambers on the plate. So you can run slightly higher, if not just one culture at a time. However, where we fit into the drug discovery pipeline is the animal risk replacement side of things. And it's basically the preclinical evaluation, the thorough analysis and the evaluation of the compounds you do before taking your compounds through the clinicals. That's where the Quasi Vivo system fit. So they are not meant for high throughput screening. However, what they are compatible with is obviously high content, because they are compatible with imaging. So we can also do fluid-based and tissue-based analysis. And as I mentioned earlier, the preclinical evaluation as well as disease modeling. So based on all of the tissue and fluid-based endpoints, there's a lot we can do which high content screening cannot deliver. Next question from Shika is how long you can culture the cells in the system.
So we can we basically cultured them up to 3 weeks or something like this, because we immediately, whenever the transwells showed differentiation, we use them for further experiments. So I mean, it could be cultured for longer, for as long as a month, or more than a month, even for 2 months actually. But we never did it for that long. We did test it with 1 or 2 transwells for longer time periods, but we didn't have to keep them in the chambers for that long, because we immediately use them for further experiments.
Thank you, Parul. Few more questions. Do you find any difference in culturing human primary cells than culturing the rodent primary cells?
Sorry, we didn't get this question. Can you repeat it, please?
Yeah, okay. So this question is from Chika. Do you find any difference in culturing human primary cells than culturing the rodent primary cells?
That's for us not possible to reply, because we only worked on human cells, and therefore I can't really reply, but I wouldn't suggest there to be a really big difference.
So just going through the list of questions. Okay, I think we've covered all the questions. So there's one question from my side. Doris, you showed in your scheme of your setup that there are different diameters of the tubings you use in your flow circuits. Can you please explain the reason why different diameters of tubes were required?
Yeah. Yes. So in the pump, as you can see, there are 2 connections in the pump. There is one tubing which leaves from the reservoir bottle. It goes through the pump and into the chambers, which are connected in series. For this, we used a tubing with a thinner diameter, and the tubing which leaves the chamber and brings the medium back into the reservoir bottle through the pump has a higher diameter. And this difference we have basically maintained to maintain a difference in the flow rate at the two tubings. And simply by using this dual pump connection, this helped in active gas exchange. And this is why we have used a different tubing. This is the first reason for active gas exchange, and the second reason is to maintain the liquid level in an early phase, because if we used, we had to do a lot of trials, and we saw that the liquid was entering into the transwells, and with this kind of a setup, we were able to maintain the liquid level at the required the required at the required level.
Right. I think there are no more questions. So we've covered off the questions. So indeed, I'd like to close this session, and now would like to thank everybody for attending and showing all the interest by posting your questions. So if you've got more questions, please let us know and get in touch with us through email to both Doris and myself. And thank you very much, Doris and Parul, as well as Simon. Thanks very much, and thank you everybody for attending. We will all get the link afterwards to listen to the recorded webinar. So thank you, Doris. Thank you, everyone.
Thanks, Bhumika.
Thank you.
Thanks, Simon. Bye.
Bye.